Hello students, I'm Dr. Arpita. A very warm welcome to my genetics class. Today we will quickly revise Mendelian genetics. This is one of the easiest part of genetics that we are going to study. If we understand the basics of it, then Mendelian genetics becomes very easy. And until and unless Mendelian genetics gets easy and comfortable for you, you'll not understand any other topics in genetics. Okay, so let's quickly know what is Mendelian genetics. We all know who was Gregor, Johann, Mendel. We all have heard about this great geneticist, right? He is known as the father of modern genetics, right? So Gregor Johann Mendel was an Austrian monk. He had started studying, he went to Vienna at the age of 21 to study science, and thereafter he started experimenting with his pea plants in his monastery garden, right? He was an Austrian monk who had studied heredity in garden pea plant, that is Pisum sativum, and published his work in the year 1865, right? Now see, two important terminologies we'll study here first. One is, see, homozygous, the other is heterozygous. Homozygous and heterozygous. What is homozygous? See, two identical alleles for a trait, example capital T or small t, small t, that is, true breeding pea plants are homozygous. Okay, two identical alleles are here. Identical means what? See, both are either capital T, both are small t. Okay, so you see it's homozygous, there is no mix, right? Next, coming to heterozygous, it is just the opposite of homozygous. Two different alleles are there for a trait. Here two identical alleles for a trait, and for heterozygous what do we have? Two different alleles for a trait, example capital T, small t. See, one is capital, the other is small, and hybrid plants are heterozygous. Here true breeding pea plants are homozygous, and here the hybrid plants are heterozygous, right? Next, see, based on his observations on garden pea, Mendel drew some important conclusions which are known as Mendel's laws of inheritance. What is it known as? Mendel's laws of inheritance. Correct? As I am teaching you in the simplest ever possible manner. Please try to take them down in your notebooks. The first law that we are going to study is law of dominance, second law of segregation, third law of independent assortment, and fourth law of recombination. Okay? So let's start with law of dominance. Correct? In monohybrid cross, it was observed that F1 offsprings of monohybrids show characters or traits of only one parent, which indicates that out of the two contrasting characters, only one appears in the F1 generation and others did not. Mind it, others did not. Thus, it was concluded that one character or factor prevents the expression of the other. Now, from the very word dominance, this word dominance has come from the English word dominant. Dominant means what? Something which doesn't allow another weaker form of it or recessive form of it to express. So here what is it being said? It is being said that out of the two contrasting characters, only one appears in the F1 generation and the other did not appear. Okay, thus it can be concluded that one character is definitely preventing the other. That is the very reason why the other character is getting masked. Okay, now see, look into this. Capital T, this is one example of a cross in parental generation. We have capital T, small t. This is dominant over this. What do we get in F1 generation? We get a hybrid which is showing capital T, small t, and the phenotype is tallness. That means what? Capital T is dominant over small t. The characters which appeared in F1 and the one which did not appear are known as dominant and recessive respectively. Okay, capital T shows dominant character and small t shows recessive character. Right? This led to the formulation of the law of dominance. See how easy it is, isn't it? The laws of Mendel are actually very easy. You just need to know the underlying meaning. If you read the lines properly, there is nothing as easy as this one, right? Okay, so next we move on to law of segregation. Law of segregation was based on the observation of the F2 generation of the monohybrid cross. Okay, so where was it? On which is it based? Law of segregation was based on the observation of F2 generation of the monohybrid cross that was performed. Members of the F1 generation are self-crossed to get F2 generation. Obviously, the members of the F1 generation, they are self-crossed to get the F2 generation, right? Mendel noted that all the F2 plants were similar to F1 and P1 generations in a ratio of 3:1. 3:1. 3 with dominant characters and 1 with recessive character. Now, what is the conclusion from this? The conclusion led to the formulation of law of segregation or law of purity of gametes. Now, what is it? See, the hybrids or heterozygotes of F1 generation contain two contrasting characters of dominant and recessive nature. Right? This is what you have already come across. These do not mix with each other but segregate or separate at the time of gamete formation in such a manner that each gamete receives only one character, either dominant or recessive. So the gametes are pure. There is no mixture of character to give rise to an intermediate form. It will either be dominant or recessive. So at the time of gamete formation, there is no mixing of the two traits. The dominant and recessive nature do not mix with each other. Instead, what happens is segregate or they separate at the time of gamete formation in such a manner that each gamete receives only one character. There is no mixing. See, if you take a look at this cross, you will understand better. See, this is the parental generation. One is dominant, the other is recessive. Okay, in F1 generation, what do you get? In F1 generation, you get a phenotypically tall plant with a genotypic composition of capital T and small t. Right? Now, what did I say? Self-crossing. See, they are self-crossed. Right? Self-crossing yields what? What type of gametes will be formed here? Capital T and small t, isn't it? There is no mix. And what do you get in F2? 3 tall and 1 dwarf in the ratio of 3:1. This is tall, this is tall, this is tall, and this is the only dwarf plant. Yes, see, this is homozygous. These two are heterozygous, and this is again a homozygous recessive. Correct? See, 3 tall and 1 dwarf plant in the ratio 3:1. This is the phenotypic ratio. Now, what about the genotypic ratio? See, 1 pure tall, 1, 2 hybrid talls, 2, 1 pure dwarf. That is, genotypic ratio is this and phenotypic ratio is this. Clear? Law of purity of gametes. Next we come to law of independent assortment. What does it say? In dihybrid crosses, okay, Mendel crossed plants with 2 contrasting characters. This one, till here you know. Plants, you had studied this in your junior classes also. So, 2 contrasting characters, plants with yellow round seeds were crossed with plants with green wrinkled seeds. Okay, in F1 generation, he obtained dihybrids which had yellow and round seeds. That means dominant character, yellow and round seeds. This means what? Dominant characters, right? Selfing of F1 plants did not yield 3:1 ratio. This ratio was obtained where? When we were studying the law of purity of gametes. This did not come. In F2 generation, this did not appear here. In a ratio of 3:2, in the ratio of 9:3:3:1, this was obtained. Okay, the new characters obtained in F2 generation neither resemble the parents nor the hybrid of F1. This is the most important line. The new characters obtained in F2 generation neither resemble the parents nor the hybrid of F1. By the observations of the dihybrid cross, the law of independent assortment was formulated. Mendel explained that the 2 characters, that is the seed color and the seed shape, are not tied together but they remain independent of each other. Right? The characters, the factors. Mendel did not use the term genes. This term was later coined. He had only talked about factors. The factors for different pairs of contrasting characters segregate or assort independently of each other at the time of gametogenesis, that is gamete formation in F1 hybrid, without affecting or diluting each other. Okay. Since we are doing it briefly here, I did not draw that checkerboard for law of independent assortment. In case you need it, you please inform me in the comment section. I'll share with you the same. Okay. Finally, we come to law of recombination. What happens here? Various stable characters appearing in several generations of organisms are determined by an association or combination of different heredity factors. Again, these factors are what we call gene today. These factors undergo reshuffling according to the law of chance. Various combination will result in the expression of various stable characters. Right? Next, along with this, I felt this should also be included, a little bit of it, not too much. Let's quickly go through linkage. Right? This is also one of the most important topics in genetics. Okay, what is linkage? The tendency of genes to pass on to the next generation in groups is known as linkage. It is linked. The very English word gives you the meaning. It is linked. They are inherited together. That is, there is a tendency or nature of genes in the same chromosome to remain together. They remain together. They don't leave each other. They remain together during the process of inheritance. They remain together during the process of inheritance. All the genes which are located on the single chromosome constitute a linkage group. So what is linkage group? What is that? The genes are located on the single chromosome constitute a linkage group. Right? The total number of linkage groups in an organism is equal to the number of chromosome pairs. Just remember this carefully. You'll understand this better. Now we'll come to the types of linkage. Broadly, types of linkage are, linkage is of two types: complete linkage and incomplete linkage. Right? Now let's take a look at what is complete linkage. When the genes for a particular trait or character are present very close to one another and are transmitted together to their offsprings only in their original or parental combination for 2 or more generations. What happens here, you see? The genes for a particular character, suppose say if you have curly hairs and this character is being passed in your family from generation to generation, like your grandmother had curly hair, your mother has curly hair, you are having curly hair, then your daughter is also having curly hair, like this. It is being passed for 2 or more generations. Okay? Let's study this with a very famous example. When ordinary male wildfly with grey body and normal wings was crossed with female having black body and vestigial wings, the F1 hybrids were all grey bodied, normal winged. The F1 hybrids had what? Grey body and normal wings. This is the dominant character. This one is the dominant character. When F1 male is backcrossed with recessive female parent, only 2 types of individuals are produced in F2 generation instead of expected 4. Right? Now these 2 types were grey bodied normal winged and black bodied vestigial winged in equal number, thus indicating complete linkage. Right? So what has happened? When F1 males were backcrossed with recessive female parent, only 2 types of individuals were found in the F2 generation instead of 4. Okay, these 2 types were what? The grey bodied fly with normal wings and black bodied fly with vestigial wings in equal number. This indicates what? Very simple. This indicates what? Complete linkage. Right? Next we come to incomplete linkage quickly. Incomplete linkage produces new combinations of the genes in the progeny due to the formation of chiasma or crossing over in between the linked genes present on homologous chromosome. Right? This is also, here it is just the opposite of complete linkage. Over there it was being passed on from generation to generation as a chunk. No break, no crossing, nothing, no mixing. Here it is different. We'll study this with the help of an example. In sweet pea plant, a cross is made between blue flower and long pollen, capital B, capital B, capital L, capital L, with red flower and round pollen, small b, small b, small l, small l. In F1, blue flower and long pollen, heterozygous condition is seen. See, this heterozygous condition is seen. Test cross between this combination, this and double recessive, this one, why double? This is there, this is there, gave blue long 43.7%, red round 43.7%, blue round 6.3%, and red long 6.3%. The parent combinations are 87.4% due to linkage, while in case of new combinations 12.6%. The genes get separated due to breaking of chromosomes at the time of crossing over in prophase 1 of meiosis, right? New combinations appear due to incomplete linkage. Just see this, I have just drawn it, I have drawn a rough diagram here to depict the cross. Okay, this is the gamete that is formed from here, this is the gamete that is formed from here, right? And this is the F1 hybrid, right? Then test cross with the double recessive. This yields this these gametes. Now see the gene combination. This is your blue, this is blue, this is red, this is red. This is what we had discussed. See, these two are equal, these two are equal. Okay, so this is how we come to an end of our discussion today. Thank you so much for your kind attention. I hope you have enjoyed revising Mendelian genetics and linkage quickly with me. If you have any confusion, any doubts anywhere, please feel free to get back to me. And stay tuned. Please like, share and subscribe to my channel. Till then, bye-bye.